Hello. Today we are going to explain the process of protein synthesis. In protein synthesis, there are two parts. The first part is transcription. The second part is translation. The C before L in transcription is why we go for transcription first, or that's how you remember that transcription is first, just for future use. So transcription. In transcription, we start with the double helix or the DNA strand, and what happens is this enzyme known as RNA polymerase breaks up the DNA and makes it into a new DNA called messenger RNA. Messenger RNA is almost exactly like DNA, but I'll get into that later. Another name for messenger RNA is mRNA. This is what I was talking about. The bases used to build DNA are adenine, guanine, cytosine, and thymine. As you see, most of them are there in the mRNA and the DNA. Except you see this purple one and the yellow one aren't in either one of them. Opposite. So you see the bases used to make RNA is adenine, guanine, cytosine, and you see a U. Well, in DNA, you know that thymine was this last one, but now it's a new molecule made of adenine called uracil. This is because RNA didn't or wasn't able to make thymine because it's a single strand and they're not exactly alike. They don't have all the properties, so they ended up having a different molecule. So now we're going to try to shift over to translation by telling you exactly what happens in the DNA. mRNA is made, and you can see the uracil and thymine are opposite. They're not together. They're a whole different molecule. But before we get into translation, I want to say that the process of transcription is to have a blueprint or starting of translation. Translation has to have something to build off of, and without mRNA, there would be no protein synthesis. So, in translation, mRNA tries to move throughout the nucleus. It leaves the nucleus into the cytoplasm using the nuclear pore in the nuclear membrane and travels until it connects to a protein. mRNA is also transformed into a protein. Once that happens, you see they're connected now. We now have this new molecule known as tRNA, or transfer RNA. tRNA and mRNA are very different. mRNA copies, sends messages. tRNA doesn't do much copying, transfer RNA, more sends things like different molecules, and I guess can piece things together. I'll talk about that later though. So on tRNA, there is things called anticodons. If you look, mRNA also has these, but they're called codons. What happens is later on, as the tRNA brings the amino acids and carries them, they will eventually go to the ribosomes and connect their anticodons, which are these, towards the mRNAs codons right here. And as tRNA and mRNA build up using peptide bonds, they start to create polypeptides, which are also known as a protein. So that is the process of protein synthesis. In conclusion, protein synthesis starts at a DNA, which is then transcribed into a single-stranded DNA known as mRNA. Don't forget, mRNA does not have thymine. DNA does not have uracil. They totally switch it around, but everything else is the same, except opposite, if you get what I mean. Anyways, the mRNA will then move out of the nucleus into the cytoplasm using its nuclear pore in the nuclear membrane, and using translation will become a protein and connect to the ribosome. When it connects to ribosome, the tRNA, which is a different molecule carrying amino acids, will then come to the ribosome and try to connect with the mRNA. The only way the tRNA can connect to the mRNA is with the tRNA's anticodons and the mRNA's codons. When they start building up to each other using peptide bonds, they create polypeptides, which are also protein. And that is a process of protein synthesis. Thanks for listening. Goodbye.